All right, great. So the last speaker of the morning sessions is our very own Dr. Hande Ozdinler. Hande received training in cell biology, anatomy, and neuroscience. She came to Northwestern 17 years ago to set up the first lab exclusively focused on upper motor neuron biology. And she's been she has a long-standing interest in defining the molecular mechanisms behind the selective vulnerability of this very unique cell type. And she's also had a long-standing interest in translating her molecular understanding into therapeutic approaches for ALS. Now, beyond her research program, as Bob noted in the morning, Hande is responsible for establishing this annual Les Turner ALS Symposium, for which we're all grateful. So, Hande, thank you.
Thank you so much. Thank you so much for this great introduction. And yes, I will be talking about the importance of building cell-based and mechanism-focused drug discovery. And I'll try to explain what I mean by that. But before I start, I always want to thank people who are working relentlessly in the Ozdinler lab, all the support that we have received. And I have many of my lab members here in the room, and they have posters. So if you are in the Ozdinler lab, can you please stand up for recognition? Thank you so much for all your hard work and for all the dedication. Thank you so much. And of course, our lab was the second Les Turner ALS lab. I was very happy to be included in the Les Turner program. But since then, we have received awards from NIH, NIA, many foundations, Department of Defense, and I'm very thankful to them. And also all my collaborators who are in this room, who are in the audience, and also joining Zoom. And I also want to thank the ALS News today. They wrote a very nice article and with the interview that they have made with me about how we think the ALS clinical trials or ALS therapies should be shaped in the future so that we can increase the success rate in ALS. So I kindly ask that you actually take a look at this, and that would summarize what I think should be done in the future so that we can increase our chances of success. And if I give an outline, I would like to tell you that in different diseases, different neurons degenerate. So there is cell type specificity, there is selective vulnerability. Not all neurons degenerate to the same degree, to the same extent. So there's very precise selectivity. And that tells us the importance of that neuron. Different neurons become vulnerable in different diseases, or because these neurons become vulnerable, different diseases occur, right? And so we need to understand the neuron that degenerates. We really focus our attention to the cells that degenerate. And then the mechanism by which they degenerate is also very different. And that's why we have to understand the mechanism of degeneration. And that's why the importance of the mechanism. And then we put these together. It's a cell-based and mechanism-focused drug discovery that is going to move us forward. So currently, what's happening is there's a drug or there's a compound that is identified for a single mechanism of action, or it binds to one thing, it does one thing in one mechanism, and it is given to a broad spectrum of patients, and some show some effect, some no show effect, some could be abnormal effect, but at the end of the day, it could be a success in phase 1, phase 2, but we have failure stories in phase 3. So then they fail. But I think if we move this to the second picture, where we know why these patients develop the disease, and we can better characterize the patients, and that we have better inclusion criteria for each clinical trial, so that we actually develop a mechanistic clinical trial, and then I think we can improve the success rate of clinical trials. So then the limiting factor is going from, if this moves, going from this to this. And again, biomarkers are needed for that. And then identifying those drugs for specific mechanisms. But I think we are getting there. We are much further along. But what are the challenges that we have? I think one of the biggest challenge that we have is the change in our concept, in our understanding, in the definition, and in the expectation that we have in drug discovery. And then what is really the translation? We can have many different experimental models. We can think that a drug enhancing the survival of a mouse is going to enhance the life of a patient, but it doesn't. So where is the translation? That's very important. And then we really need a scientific matchmaking, which means this patient would respond better for this drug, this patient responds better for this drug. We really have to form that scientific matchmaking. And then we should accept the fact that Godot is not going to come and that one drug is not really going to cure all problems, because we are really fighting with a beast. But I want to remind you that FDA was established in 1906, and the first IND application was made in 1962. And at the time, FDA, all the rules and regulations, all the expectations, the FDA language, everything was written before 1962. And those have not been greatly changed. And at that time, we didn't have proteomics, lipidomics, metabolomics, all these gene expressions, like we did not have any of that. But we had clinical observations, and that, the clinics were very well defined. So, the patients and the diseases were defined by what the doctors very carefully, described, but without any cell biology or molecular biology information. So then the language that FDA uses is based on the data that we had in the late 1800, 1900. That's very old. And that at the time, FDA suggested that each disease was a distinct entity. So each circle here represents a disease, and that they said diseases are distinct entities, and that the goal is to cure each disease, okay? So they thought that these diseases were different. But now we realize that diseases are like branches of the same tree, and that many of the diseases have so much in common. So it's like the pictures like this. It's not like different circles do not touch each other. It's actually a massive neurodegeneration problem. There are so many common aspects of neurodegeneration, and yet there are distinct different branches. So in the past, the idea was that diseases are different and patients are the same. But now in 2025, we realize that patients are different and diseases share so much in common. That is a huge difference of what FDA was thinking and how the rules were made. And now we are trying to fit into the rules that were written, I don't know how many years ago, but the reality is different now. And that's one of the reasons that we continue to fail. If we cannot change our perception, we will not be able to find drugs, because patients are different and diseases share so much in common. That's the new reality. And of course, we are working with the brain, one of the most complex organs in our bodies. This is what Ramon Cajal has drawn, like more than 100 years ago. He realized the heterogeneity of the brain. It's not that different regions are different. Even within the same region, there's heterogeneity. And then ALS has component both in the brain and in the spinal cord. Like different neurodegenerative diseases such as Alzheimer's or Huntington's, Parkinson's, they are still within the brain. ALS is the only one that has both cortical and spinal components. So it makes it even more complicated. Okay? And again, the question of who dies first, is it the brain and then the spinal cord? Is the spinal cord then the brain? I wish we didn't have those conversations, because it is the circuitry degeneration. London Bridge is falling down. You don't care if it is this leg or that leg. The whole system is falling down. And that makes it even more complex. And now complexity is added to the understanding of the disease mechanism. Now it's not just one mechanism, it's the mitochondria, it's the protein aggregation, it's the ER, it's the granules, it's the axon transport, it's the excitotoxicity, it's immense. It's like a beast with 100 arms. How are you going to fight with this beast? You can target your drug for one arm. He has 99 left. And there's also intrinsic factors. There are also extrinsic factors, astrocytes, microglia, other non-neuronal cells, the environment, it's a huge problem. And if you think about this, the connection with the muscle, the connection from the cortex to the spinal motor neuron to it is an immense problem. How are we going to find one drug that is going to kill all those 100 arms at the same time and very effectively? That's why we fail, because it's very hard. And that one drug may not come, Godot may not come. So then people begin to think about what is common. At least let's focus on the things that are common, right? And then we begin to realize that there are so much in common, actually. If you think about other neurodegenerative diseases, all neurodegeneration, protein aggregation is actually a very good start. There are many proteins that aggregate. Even though the identity of the protein is different, the protein aggregation problem is common. And, there is axon transport problems, cytoarchitectural problems, mitochondrial problems. And the protein aggregation problem is actually a good target, I think, because at the end of the day, the proteins aggregate. And if we can eliminate the aggregation, and if we can stop the aggregations to occur, that may be an effective treatment strategy, not just for ALS, but maybe neurodegeneration. Maybe it will be an important arm of the tree. And the design of the clinical trials is very strange. First, there's this preclinical stage. As we know, the scientist is a very long road, because you have to identify what is going wrong, and you have to identify the solution. You have to confirm with this method, that method. I mean, it can take years. It can take 10 years, 20 years. It's huge, and it's very expensive. But let's say you have some compounds, and then phase 1 is for toxicity, trying to see if this is toxic or not. Like, is it good and the patients can actually tolerate it? Small number of patients, and most of the drugs that fail later on actually pass phase 1. So then, they're okay. Phase 2 is efficacy, which means does it work? But then to assess whether it works or not, you need some very strong fiduciary markers. You need something that would tell you quantitatively that it is working. So you need biomarkers. You need a very nice assay to see engagement, to see response. We don't always have that. What is the biomarker? What are the markers that distinguish different patients? How are we going to know which compound will be effective? Those markers may also be dependent on which compound you try, right? So that knowledge is a bit lacking, but it is coming. It is coming. Phase 3, I think, is a dead sentence, because then you actually include so many patients in the study, so much heterogeneity, and now you expect Godot to come and that magical drug to cure everyone. And that's exactly the phase that most of the clinical trials fail. And I think we really have to understand who would be, responding to treatment before we start phase 3. And even for rare diseases, maybe the definition of phase 3 could be different. Again, let's come to the challenges. Where is the translation? So work in our lab and also in other labs show that the translation is at the cellular level. What do I mean by that? If you compare mice to human, there are different species, and mice survival does not translate to human survival. But if you look at the neurons that degenerate in well-defined model systems and the motor neurons in patients, at a cellular level, they're almost identical. The problems that they share are identical. They have, for example, mitochondrial problems, protein aggregation problems, cytoarchitectural disintegration problems, nuclear pore membrane and nuclear transport problems. Everything is the same. And if I were to show you two slides, one from the human, one from the mouse, upper motor neurons, you couldn't tell which one is which. So I think that focusing our attention to the neurons that degenerate could be translational. And work from Dr. Gautam, he's in the audience, and also Baruch Genchen, I think he's in the audience, show that looking at well-characterized disease models, upper motor neurons, and if you compare them to human, they are exactly the same. And that's why we generated the first upper motor neuron reporter line in which there is a GFP expression restricted to the upper motor neuron. And that was such a big deal because that was the first reporter line very specific to a distinct neuron type in the motor cortex. And that's why we were funded. And, now we actually share this mouse model with other researchers at Jackson Lab. You can order this mouse model. And that allowed us to switch our focus from mice to neurons that degenerate. And that's why we cross them with so many different disease models so that we isolate or we have upper motor neurons that degenerate due to different underlying causes, and we use pure populations of them by performing FACS, fluorescence activated cell sorting. And after that, we do proteomics, metabolomics, RNA-seq, to interrogate exactly what is going on in these disease neurons at different stages of the disease and in comparison to other underlying causes of the disease. As you can imagine, this has been an immense undertaking in our lab. We are not fully completed, and we spent maybe more than $1 million so far for this project. But I think it's going to reveal at a cellular level the basis of selective vulnerability. Here I want to show you that we have shown in the SOD1 mouse, and this is P15, and P15 is postnatal day 15. They have not yet opened their eyes or are just beginning to open their eyes. There is no disease, but at a molecular level, they begin to show defects. And, the lipid that was actually mentioned this morning, there is dysfunction at lipid. And then there are cytoarchitectural problems, mitochondria problems, there are protein aggregation problems, there is now astrogliosis, macrogliosis. As the disease progresses, also the signature of events are changing. And combining this with proteomics approach, RNA-seq with proteomics, now we begin to reveal what are the changes in the protein. And that was actually the grant that we received from DOD, together with the disease models and blood samples obtained from ALS patients. We put this together, try to understand the protein profile and the changes in that profile as the disease progress in different disease models, and compare it to the human samples. And when you compare this data from human and mouse, believe me, most of the proteins that are important will be conserved among species. So then we begin to understand what could really be important for the disease progression, and some of those will be informative about biomarkers and pharmacokinetic biomarkers. This is our paper, just recently came out. Dr. Gautam is the first author, and I think he has a poster outside, that we have isolated exosomes from SOD1, patients with SOD1 D90A mutation that's showing upper motor neuron degeneration, and applied proteomics to those exosomes. And Dr. Savas is in the audience. Thanks to him, we were able to perform this study. And by studying the proteins in the exosomes, we begin to identify some potential biomarkers and pharmacokinetic biomarkers. This is just tip of the iceberg. We have not yet published any of our results, and those results are coming. But when we realize that, identifying of these neurons in making them fluorescent and asking them, it actually got the attention of drug discovery today. We were invited to write reviews about this, that when we want to move forward with clinical trials, we really have to get the information from diseased neurons, and that we have to incorporate their survival requirements in our clinical trials. We can't be using with fibroblast cells, who cares what cells have nothing to do with ALS. We really need to ask the neurons that degenerate and get exactly the answers, the exact answers from them. And that's why we actually are developing a drug discovery platform where we use diseased upper motor neurons. And that was also the grant that we received from DoD. And Dr. Quintanilla is in the audience, and he has been instrumental in developing this platform so that we can investigate directly whether the compounds of interest improve the health of diseased upper motor neurons. But how about patients? Do we always use mouse to understand ALS? How about patients? I think patients really tell us about the disease, and every patient has secret information in them that we have to find a way to reveal that. And that's what we did. So far, there were 147 genes identified in ALS patients. And if you look at those genes, they seem unrelated. They code for unrelated proteins. But then, when you ask the question, which other proteins do these gene products bind to, and what canonical pathways are they in, and what games do they play in the cell, they begin to inform us. And this was actually made news in Science Trends, that human mutations are so informative that we can actually begin to understand disease mechanism. And yes, indeed we could. Because when you put all these proteins together and say, proteins, tell me, what are you doing? And they say, well, I really try to make sure that I maintain homeostasis, but if there's this mutation in that gene, everything falls apart and I cannot maintain it. And, it's really important that I keep the cytoarchitectural lipids and, the proteins and so forth. And I said, okay, any one of you are more famous than others? Do you build games, like you do build bridges, and who talks to whom? And say, yes, this guy is very famous. He talks to everyone. He interacts with everyone. And if there's a mutation in that guy, we all fall apart. And then, all right, so how is that this guy expressed in healthy neurons and diseased neurons? So imagine this, even though there is no mutation, look at this. The proteins aggregate, and they are in those clusters, and there is no mutation, but the protein is not able to do what it is supposed to do. Sometimes you don't have to have a mutation to have a defect, right? So I may be sitting in my office, but that doesn't mean I'm working, right? And proteins may be in the cell, in the soma, but if they are not working, who cares if they're in the soma? So then where they are and if they are doing what they are supposed to do is really very important. And, we published this recently and then looked at the canonical pathways, what they are doing and which proteins are involved and what are the secreted proteins and so forth. We did the same thing for HSP, PLS, and we also looked for the spastin and alsin protein, their binding partners and so forth. And come to the defining of the problem that we realized after putting all this data together, upper motor neurons suffer 4 major things. 1, protein aggregation. 2, mitochondrial dysfunction. ER stress, disintegration of the ER. Cytoarchitectural problems, axon transport defects. Those are their major problems. So then that's good, because then we actually have an idea of how to treat them or develop treatments towards them. And my collaboration with Dr. Silverman, and he's also in the audience, has been very productive and fruitful, because he's one of the most well-known medicinal chemists, and he at the time generated lines of different compounds reducing misfolded SOD1 using different cell lines. But working together, we realized that the compounds that he generated actually reduces misfolded SOD1 both in vivo But then we realized that these compounds also help reduce TDP pathology, which was like a different ball game, and the compounds were not identified based on TDP. We also put them in culture together with riluzole and edaravone, and then there were also edit effects. It speaks to the volume of the mechanistic insight because the mode of action is different. So then, when you actually give a NEO9 to diseased upper motor neurons, to be honest with you, I was expecting maybe it's going to do one good thing, right? But we have seen reduced protein aggregation, improved mitochondria, improved ER, and improved cytoarchitecture. In my opinion, this is the first that a compound can do 4 major things, and that's why we are very excited about this, that a NEO9 treatment actually reduces the SOD1, misfolded SOD1, and this was actually a study done by Barış Genç, I think he's in the audience, and that if you look at the TDP pathology, and this was actually done, work done by Emel Ulupınar, and I think she's in the audience, that in the TDP mice, and these are transgenic TDP mice, they have ubiquitin accumulations, but with TDP treatment, you actually don't see that anymore. I'm going to go back. Yeah, so then there is reduced, almost like, you clear ubiquitination in the TDP. But most excitingly, okay, so this was in the context of ALS. But Dr. Silverman, in collaboration with Dr. Klein, tested this NEO9 with A-beta amyloids within the context of Alzheimer's. Now, this is a totally different ballgame, but it's the same mechanism, protein aggregation, as the common problem, and they were able to see that NEO9 also reduces A-beta gamma. And I mean, that I think is very interesting. We looked at the 5XFAD mice in the hippocampus and in the motor cortex. And 5XFAD mice is a mouse model for Alzheimer's, but they also have protein accumulation problem. And guess what? NEO9 actually reduces that in the hippocampus and in the motor cortex. So this was work done by Barış and Emel in the lab. But that speaks volumes to the fact that if you focus your attention to the mechanism such that the common mechanisms of neurodegeneration, there may be better hope. And Dr. Silverman created the company Akava Therapeutics, and we received $7.3 million from NIH to further investigate NEO9 within the context of neurodegeneration. And within this climate, I think this award speaks volumes to the importance of this work. And NEO9 was identified based on an in vitro protein aggregation assay on misfolded SOD1, but we found that it reduces the levels of misfolded in vivo. It reduces beta amyloid of 5XFAD mice, ubiquitinate proteins in the motor cortex, also in the TDP mouse model. So then different proteins, but NEO9 can help eliminate them. And then it has potentially an overarching effect. So then it's not restricted to one protein. I think it may be solving the mechanistic problem in general. So then if I come back to this very complex picture of ALS pathology, and then our need to move from the first part of this figure to the second part, I'm sorry. Sorry about that. I think it's really important to understand that our goal is not really to end the disease. Our goal is to improve the health of the neuron, so that by improving the health of the neuron, the motor neuron circuitry will improve. And by improving the motor neuron circuitry, the clinical outcomes will be improved. And because the clinical outcomes will be improved, the patients will feel the relief. And when patients feel the relief, then the disease will be eradicated. So we don't expect to eradicate the disease before curing the patient. So the goal is the patient, and the patient is the center of the attention. And that's what also is the motto at the Les Turner Center and the Les Turner Foundation, that the patient is the center. And I think that's how it should be moving forward. And I really think that there's a bright future ahead because we understand disease mechanisms every day we understand disease mechanisms much better. And that if we do this scientific matchmaking and if we generate better clinical trials, include potentially improving patients to our clinical trials, and maybe develop personalized medicine approaches, there will be a bright future for everyone. So I thank you for your attention, and I'm ready for your questions.
All right, questions for Hande. Bob.
Hande, I just want to read a question. I'm quoting, "I've been concerned that if we keep getting the wrong answers, perhaps we're asking the wrong questions. So what do you believe? For example, what should the FDA be doing, should be asking? What should the FDA be asking?"
Of course, that's a tough question. I don't know what the FDA should be asking, but I think the question that needs to be answered is what is the major mechanism that becomes dysfunctional for that particular patient? And then what is the drug or compound out there that would address this mechanism? And can we again do this matchmaking so that we understand the disease mechanism and mechanism of action of compounds and then find the right patient population for them? I think, FDA may facilitate that, and we really need to be more clever in our clinical trial design. But for example, FDA, the first question they ask is, which disease are you after? Rather than which disease are you after, it should be maybe which mechanism of disease or which is the underlying cause of the disease that you are after. Maybe in the future we may have basket trials, combinatorial trials, as long as patients suffer from the same or similar underlying conditions. Yes.
Adrian.
Hande, what do you think is the mechanism of action of NU9? You sort of told us we shouldn't wait for Godot, and now you're telling us that this molecule does everything. So
yes, This is a very good question. It's a $10 million question. So, Dr. Silverman may answer more, and, they have table outside. We can ask as well. But in my opinion, where does it bind to is a detrimental question, that it may not be binding to one thing and does one thing. So, we have done CETSA studies, we have done protein-protein interaction analysis and so forth. So I don't think that it binds to one thing. That's what my understanding. And the other thing is there are proteins that are important for maintaining homeostasis. And that homeostasis also includes, maintaining, like, for example, Yvette's talk, maintaining, for example, mitochondria-ER interaction, its association with TDP pathology, right? So at the end of the day, everything is actually linked together. And there are some proteins that are on top of the food chain. It may be that NU9 modulates their stability or modulates their interaction with another protein and facilitating, membrane dynamics, membrane stability, and cytoarchitectural stability. For example, if you have one of the things that we know NU9 does is improve the inner mitochondrial membrane integrity. Once you do that, you actually solve many of your ATP problem. You generate a lot of energy. And the other thing is ER membrane falls apart in ALS. With NU9, you also restore that. And that tells you there's reduced ER stress, there's better quality of production of protein. And, clearance of the protein, the autophagosome pathway is also important. So, you can address it in both ways. Either clear the proteins that are aggregated in a much faster way, or also eliminate or slow down the production of protein aggregations. So I think NU9 could be doing multiple things.
So Hande, can I ask you a question?
Of course.
One of the things that I'm wondering about is a lot of the work that has come into central focus of the last few years in relation to TDP-43 loss of function effects, splicing alterations, argues that there are a host of probably critical or physiological splicing errors that do not occur in mice. They seem to be either human or primate specific. What are you concerned at all about our ability to model those aspects of the disease with animals?
Yes, this is a very good question, because especially because TDP is a protein that is important for splicing of messenger RNA, and that splicing machinery may not be exactly identical in the human versus mouse. And especially if you're working on TDP model and the question that you're asking are within the context of RNA splicing, then you may miss some of the genes that are spliced in human, may not be spliced in the mouse. They might not even have that gene. So I think for questions that are related to that, mouse models may not be the best. But for cellular pathologies, let's say mitochondria problem, ER problem, cytoarchitectural problem, like if those are your readouts, then they are feasible. But if you're really looking at individual or specific genes that are spliced this way, that way, maybe cell lines or iPSC cells may be better.
Don.
Yeah, so back to NU9, I think Adrian raised it, that you can fail to accumulate or stop accumulating very different kinds of aggregates, intracellular and extracellular. Really, that's just really unexpected. So I think I kind of disagree with the statement that you made, which is we don't care how it works. I think we do care. We care a great deal of how it works, and it's really confusing. A beta is outside the cells, and the others are inside the cells. How you would with a common agent, how you would manage that is really perplexing.
I agree with you, and if I said I don't care about how we understand how it works, I don't think I've said that. So we do want to know how it works. But I don't think that it works by just binding to one thing and doing just one thing. So it may be that, you are the best person to show us the involvement of non-neuronal cells, like the neighborhood, astrocytes, microglia, right? So for example, we have an MCP1 CCR2 model in our lab to look for the astrogliosis, microgliosis, and its impact on motor neuron survival and death. So it may be that when we give the drug, we only look the neuron survival, but that compound may have an effect on the astrocytes, microglia, other cells in the environment, which we don't study with cellular precision that we do with the upper motor neurons. So I would imagine that when we take, let's say, a compound or a drug, even though it is geared towards one neuron type or one cell type, there are many other cells that are in support, or they are part of the circuitry. There is something going on in them as well. So, as you said, having that finding from a third lab and with Dr. Klein and within the context of A beta and extracellular, and I think that's the reason why we received the grant to further investigate how is that possible and feasible. And hopefully within 5 years, I will be able to give you a better answer. But I agree with you. This is the first time that I see a compound which can actually address multiple problems at the same time. And I think as we move forward, we either will have combinatorial treatments using different drugs that are addressing precise problems, or use compounds like NU9 or others like NU9, which address multiple problems, but one compound who could do this. But exactly, understanding how it does, I wish I knew, and I'm going to spend 4 or 5 years trying to investigate that.
All right, great. So let's continue the discussion during lunch.
Thank you so much.
Thank you, Hande, again. And we have a long lunch. We'll reconvene at 2:15.